I have divided the adaptive immunity pre-work material into 3 short mini lectures. You'll see the objectives listed below for the entire adaptive immunity module. This first mini lecture will focus on T-cell activation. The outline for this lecture will be to discuss dendritic cell activation, the signals that drive the activation of T cells, including signals 1, 2, and 3, some of the key events of TCR signaling and clonal expansion, the effector differentiation of activated T cells, and the role of follicular helper T cells. So I'm going to draw your attention to a slide I shared previously, which shows a macrophage and a dendritic cell within tissues that's recognizing a pathogen. In this case, it looks like some bacteria via expression of their toll-like receptors. I want to focus today more on the dendritic cells. And when dendritic cells recognize this pathogen via their toll-like receptors, it induces their activation. In the skin and in other tissues, they live in an immature state. But when they are activated, they migrate to lymph nodes where they can then activate T cells. There are 2 different ways in which dendritic cells can become activated. One is the recognition of pathogens directly via their toll-like receptors. A second is that dendritic cells can be exposed to cytokines in their inflammatory environment. This includes IL-1 and tumor necrosis factor, and IL-6 also includes, in the case of viral infections, type 1 interferons such as interferon alpha and beta. When the dendritic cell becomes activated, it undergoes a number of important changes. The first of those is that it increases its ability to be an antigen-presenting cell and to activate T cells. And it does this by increasing the expression of MHC. It increases the expression of a class of molecules called co-stimulatory molecules, and it induces the production of cytokines such as interleukin 12, all of which are important for T cell activation, and which we'll discuss in more detail in the coming slides. They also change expression of certain chemokine receptors, such as CCR7, and this has the effect of causing these dendritic cells to migrate out of the tissue and towards draining lymph nodes via lymphatic vesicles. Also, free antigens and pathogens can migrate from these tissues and drain from these tissues into the draining lymph node. Once dendritic cells migrate into tissues and begin presenting antigens to naive T cells, they can induce the activation of those T cells. And there's a couple of important things. First, I'd point out that this interaction between a T cell and a dendritic cell can last for hours, even up to a couple of days. And part of the reason for this is that this interaction is stabilized because activated dendritic cells express a lot of adhesion molecules, such as LFA1 or ICAMs, and these adhesion molecules make them very sticky and allow the T cell to form these long-lived interactions. Once T cells become activated, and sometimes this is called T cell priming, it induces proliferation and cell division of these T cells called clonal expansion. And as these cells clonally expand, they also develop effector functions. And so often activated T cells that are mediating antipathogen functions are called effector T cells. And 2 examples of effector T cells are helper T cells and cytotoxic T lymphocytes. For these T cells to get activated, they have to receive 3 main signals. Signal 1 comes through the T cell receptor. Signal 2 comes via co-stimulatory molecules. And signal 3 comes via cytokines that induce effector differentiation of these cells. So first, we're looking at a dendritic cell expressing MHC class II and a T cell expressing a T cell receptor that is specific for this foreign peptide being presented by the MHC molecule. And this is signal 1. But T cells don't have an intracellular signaling domain, so they require other accessory molecules to mediate signal 1. The first of these is CD4, or if it's an MHC class I, it would be CD8. And this is called the co-receptor. And co-receptors stabilize the binding of a T cell receptor to a peptide-presenting MHC molecule. And these co-receptors also associate with tyrosine kinases in their intracellular domain that bring them in close proximity to the T cell receptor and helps initiate T cell receptor signaling. The other part of signal 1, another accessory molecule, is a group of molecules that cluster together that are collectively called CD3. CD3 is composed of a number of different subunits. These subunits, because the T cell receptor doesn't have signaling domains, these subunits mediate the TCR signal. And each of these subunits in their intracellular cytoplasmic domain contains multiple activation motifs that allow for phosphorylation of tyrosines. These are called immunoreceptor tyrosine activation motifs, or ITAMs. And phosphorylation of these activation motifs initiates a tyrosine kinase signaling cascade. Okay, so that's signal 1. Let's move on to signal 2. Signal 2 is mediated by co-stimulatory molecules. And while there are a number of co-stimulatory pathways, we're going to focus on the B7 co-stimulatory molecule. There are actually two versions of B7, B7-1 and B7-2. And B7-1 and B7-2 bind to a molecule on on all T cells, I should say, called CD28. And CD28 in turn sends a survival and activation signal to the cell. So activation of dendritic cells is what induces the expression of these B7 molecules. Naive T cells always express CD28, and again, this binding delivers an activation signal to the T cell. As I mentioned, there are multiple co-stimulatory pathways. For the purposes of this class, B7 and CD28 are the pathway that I would like you to be aware of with regards to T cell activation. And it should be noted, co-stimulation is required for productive T cell activation. If you get signal 1 in the absence of signal 2, instead of activation, you get T cell tolerance, or the T cell may even die. Okay. Now, in addition to these co-stimulatory signals delivered by CD28, there are also negative co-stimulatory signals delivered by a pair of molecules, CTLA4 and PD-1. And both of these molecules are not expressed on naive T cells, but they are upregulated within a few days after T cells get activated. CTLA4 also binds to B7-1 and B7-2, but it competitively inhibits CD28 from binding to these molecules. And it does that because it has higher affinity actually for these molecules than CD28 does itself. Both CTLA4 and PD-1 induce a negative co-stimulatory signal within the T cell that suppresses T cell activation and attenuates TCR signals. And one way to think about it is that CD28 is the accelerator, but in order to control T cell activation, you need brakes. So PD-1 and CTLA4 are the brakes on T cell activation. And they're important because when you get rid of PD-1 or CTLA4, one becomes susceptible to autoreactive T cell responses. And so these are important mechanisms for maintaining tolerance and preventing T cell mediated immunopathology. Also, I should mention, and we'll talk a little bit about this more in our cancer immunology lecture, but CTLA4 and PD-1 are often referred to as checkpoints on T cell activation and are now targets for cancer immunotherapy. The last signal that I wanted to mention is signal 3, and this is induced by cytokines. So here we have B7-1 and B7-2 binding to CD28. We also have upregulation of these negative co-stimulatory pathways to kind of control this response. So signal 2. CD3, CD4, and the T cell receptor deliver signal 1. And I mentioned previously that there's a number of adhesion molecules like integrins and ICAMs that are upregulated during this process that mediate the interaction between the T cell and the dendritic cell. But dendritic cells, when they become activated, also begin to produce inflammatory cytokines. One of those key cytokines is interleukin-12. As one example, and what I'm showing here is interleukin-12 binding to the IL-12 receptor on T cells and inducing T helper differentiation. So these signal 3 cytokines can be produced by the antigen presenting cell, but they can be supplied by other cell types. And the key function of these signal 3 cytokines is to induce the acquisition of effector functions. Now I'm going to take a moment to discuss T cell receptor signaling. And this slide looks a little busy. I don't want you to worry about it too much. There's just a few key points that I want you to take away from this. But I did want to take you through the whole process of how a T cell receptor initiates a signaling cascade that results in cellular activation. So here we have CD3 subunits that are associated with the T cell receptor. When the T cell receptor binds to its antigen, it draws CD4 into proximity with these CD3 subunits. CD4 constitutively associates with this tyrosine kinase LCK, and when it comes into proximity of CD3, LCK begins to phosphorylate all of these activation motifs on these CD3 subunits. And this phosphorylation in turn results in the recruitment of other tyrosine kinases like ZAP70 and tyrosine kinases that induce activation of the MAP kinase pathway. So what this results in is a tyrosine kinase cascade that results in the activation of a pair of transcription factors, AP1 and NF-kappa B. ZAP70 further goes on to activate phospholipases that in turn induce the influx of calcium either from the extracellular space or from the endoplasmic reticulum, which is a major store of calcium. And when calcium enters into the cytoplasm, it activates calcineurin, which in turn activates this transcription factor NFAT. And together, these three transcription factors, AP1, NF-kappa B, and NFAT, induce T cell clonal expansion, differentiation, and survival. One thing I do want to note, remember this calcineurin and NFAT pathway when we come back to discuss immunosuppressive drugs that are used during transplantation. Calcineurin inhibitors are a major class of immunosuppressive drugs in that setting. So NF-kappa B, AP1, and NFAT, when they become activated, translocate to the nucleus and induce the expression of many genes. I'm going to focus on only two of those genes initially. And those two genes are interleukin-2 and the interleukin-2 receptor. T cell activation results in T cells rapidly producing lots of interleukin-2 and secreting it into the extracellular space and expressing the high affinity version of the interleukin-2 receptor. When this happens, interleukin-2 can bind to the interleukin-2 receptor either on the same cell that secreted the IL-2 with autocrine stimulation or on neighboring cells in a paracrine fashion. And when that happens, IL-2 drives clonal expansion. These T cells start to divide rapidly. And so we get clonal expansion. And to give you an idea of the scope of that, one activated T cell can give rise to up to 10,000 to 100,000 daughter clones, all with the same TCR specificity. So this is the way we go from T cells at very low frequencies to T cells at very high frequencies that can mediate clearance of a pathogen. Now, I mentioned that signal 3 induces effector T helper subsets, and this shows a dendritic cell presenting antigen to a T cell delivering signal 1, as well as costimulatory molecules delivering signal 2. This dendritic cell may produce cytokines that induce further differentiation of this T cell, or they may receive signal 3 cytokines from other sources. And this results in the formation of 4 main subsets of helper T cells. The first of those subsets are Th1 cells. T helper 1 cells are induced by the signal 3 cytokine interleukin 12, and T helper 1 cells specialize in the secretion of the cytokine interferon gamma. The second of these subsets are T helper 2 cells. They are induced by the signal 3 cytokine interleukin 4, and T helper 2 cells in turn secrete interleukin 4, interleukin 5, and interleukin 13. Th17 cells are induced by IL-6 as a signal 3, and Th17 cells in turn specialize in secreting the cytokines interleukin 7 and interleukin 22. And lastly, the signal 3 cytokine transforming growth factor beta can induce the formation of regulatory T cells. And when this happens, these regulatory T cells secrete immunosuppressive cytokines such as interleukin 10 and transforming growth factor beta that suppress the activity of all of these other subsets of T cells. So we've discussed each of these subsets. And so when T cells get activated in the T cell zone, by dendritic cells expressing antigen, it can lead to the formation of Th1 cells, Th2 cells, and Th17 cells. Now, We'll talk in the next mini lecture about the effector functions of these cells and what they do. It's important to note that sometimes an infection can induce multiple subsets. So you can get Th1 responses and Th17 responses together, for example. And when these effector subsets are induced, they migrate to inflamed tissues. But there's a subset of all of these responses that instead of migrating to inflamed tissues, they express a chemokine receptor called CXCR5. And this induces these cells to migrate to the B cell zone. And for that reason, these cells are called T follicular helper cells, because these are the subset of cells that specialize in enhancing B cell activation within the B cell zone. So when we talk about B cell activation, we'll come back to the role and function of these T follicular helper cells. Lastly, I did want to mention that CD8 T cells also require signal 3 for their activation. And the signal 3 for CD8 T cells is interleukin 12 or type 1 interferons, interferon alpha and beta. And these cytokines induce CD8 positive T cells to differentiate into cytotoxic T lymphocytes. These cytotoxic T lymphocytes can secrete inflammatory cytokines like interferon gamma, and they also secrete cytotoxic granules that can kill virally infected target cells. CTL responses don't always happen in isolation. They're usually accompanied by T helper 1 responses, which makes sense given that the signal 3 cytokines for Th1 and CTL responses overlap. So here's a summary of some of the key points from this mini lecture. Please take a moment to go over each of these points to review the material that you learned today.